Hi, everybody. Thanks for joining us tonight. I'm going to be presenting Casting a Net for DNA, our Carp Barrens Part 1 presentation. So in October of 2021, Janet Mason and I funded a small informal pilot to do a DNA-based inventory of 3 ponds in the Carp Barrens. Mary Gwen Miltenberg, a biologist, joined us to perform the water sampling. So the presentation today is about environmental DNA, also known as eDNA, and our initial eDNA findings. So many of us have heard that the Carp Barrens area is really biodiverse, but how do we know this for species that are really hard to find, see, or touch, or hear? Can we prove it? Well, we can now, and there's this really amazing technology that helps us do this. And this is what this presentation is all about. So a few assumptions, and a bit of an overview first. So I'm just going to talk a little bit about my background very quickly. Assumptions and goals, quick biology 101, very, very quick. The results we found out, the next steps, and how you can get involved if you're interested. So just a few assumptions that I'm making about you since you've joined us tonight. So the first one is that you're curious. You know, you've looked at a bug or a tree or a leaf, plant, or animal and wondered, what is this? And maybe wondered, what is its place in the ecosystem? You believe biodiversity is important, so the totality of genes, species, and ecosystems of a given region. And maybe you believe, like I do, that biodiversity is an important measure of wealth. I am also assuming that you know what DNA is in a general sense, but you're unfamiliar with eDNA. Now, if you're a biologist, I have a lot more technical slides and reference materials I can share in the future. So I read between 50 and 75 papers as I journeyed into this interesting world of environmental DNA, also known as eDNA. And there are a number of considerations for serious studies using eDNA. So the things that I'm hoping you'll learn from this presentation is I just want to make you aware of eDNA and its potential. It's an important technique and it's emerging, and it's partly driven by reduced costs in sequencing and advances in bioinformatics. And there's also many industry and academic efforts to catalog all the life on the planet using eDNA. And some of them are happening, out of University of Guelph is a big part of that, and other academic institutions in Canada. I want to show you that we have significant biodiversity in the Carp Barrens, outside of what we already know we have that's connected by or collected by naturalists, for example. Want to interest you in supporting formal eDNA studies in the future. It has zero impact on the environment, and the information it generates can help us become better stewards and defenders of these places that we love. And I want to give you some simple ways to learn more and get involved. So I'll give you some of those at the end of the presentation. So first, let's just get situated in the Barrens. If you're joining us from another remote location, just fly in here with the friends from Google Earth, and you'll see quickly that the Carp Barrens is located, we've got Canada over here, we've got Carp here, and then the Ottawa River over here. And the Barrens right in the middle of all that dense urban and farming area. So just quickly, a little bit about me. That's a pond near my house in April 2021. I think there's 19 turtles on that log and at least 2 blanding turtles. I think there's one here with the yellow, distinctive yellow neck, and one there. And a couple pictures that I took with my microscope. A few years ago, I was reading this book, The Gene and Intimate History, and it kind of rekindled my interest in biology, which, to be honest, I hadn't thought too much about since high school. I went out in the middle of winter, I chopped a hole in the ice, and I looked at the water samples under my microscope, and I looked at these things, and I think that's protozoa. And I wondered, okay, what are these things? And, is there maybe a different way that I could learn about them without, taking pictures of images and looking them up. So I was doing some research and reading, and I came across this new sequencing technology, the nanopore sequencer, and I was really excited about it because it was around $1,000, and I could sequence the DNA of these things, and I could kind of impress my friends and impress myself that I was learning how to sequence DNA. But I pretty quickly learned that buying $1,000 sequencer is like buying a car, and you need to manufacture your own gas, where DNA is like the gas in this instance. So I haven't lost faith in that. I'd like to do that, but for now, I focused on environmental DNA. Kind of in that period, though, Janet introduced me to a professor from Carleton University, Dr. Sue Aiken, who kind of asked an important question, are you more interested in doing the science or finding the results? Meaning, do I want to spend $20,000 or more to extract my own DNA and maybe get it right, or maybe not, or leverage an existing service right away and gain some answers and contribute to our knowledge? So I found my way to environmental DNA, and right now I'm taking a course at University of Guelph on DNA barcoding. So trying to further my understanding. So just to kind of frame the mental model here, many of us, we have gone to a pond or stream or lake, and we've scooped out some water with a bucket or net, and we've looked at the contents and wondered what is there. Like this little girl, she's thinking maybe about, minnows. Well, eDNA is like dreaming, catching minnows and discovering everything that's interacted with the pond in the last 7 to 21 days. So frogs and turtles and insects, plants, bacteria, birds, coyotes, beavers, and even dogs in this case. Because every living organism sheds DNA, and some of it ends up in the water. So environmental DNA it's almost like magic in terms of the wealth of information it provided to us. But first, a very, very brief biology kind of reminder. So DNA is found in all living or once living things. So it's a molecule that's common to, animals, plants, bacteria, and it contains genetic information that's unique to each individual. So we all know that we have this DNA signature, and because of the pandemic, we know that DNA can be picked up by really uncomfortable mechanisms like nasal swabs. Well, eDNA just picks up naturally what is being shed at all times by people or other organisms. So no nasal swabs or other invasive means. And what we're doing is we're targeting specific gene regions for these nucleotides, so these A, C's, G's, and T's. So we're using some special techniques and equipment to do that. So normally when you're doing aquatic biodiversity, so you're doing some kind of inventory, the traditional methods are like electrofishing and kick netting. So you're doing this for streams, right? So you zap whatever's a fish is going by, you catch it in a net, you tag it maybe you take a little tissue sample. And the other way is the kick netting, where you're standing upstream, you disturb the bottom of the stream, and you collect stuff into a net, and then you count it, look at it under a microscope, perhaps. eDNA is much more gentle, really. You're basically collecting water, pushing it through a filter, and then sequencing the DNA on that. So conceptually, eDNA sequences are like nature's barcodes. If you think about the products we buy in the supermarket, they have this barcode and it translates into a product. Well, you might have this barcode here, which translates into a DNA sequence. And all this information is stored on databases, many of them publicly available. And if you had your phone out and you had a QR code reader and scanned it, you should end up with this sequence of letters, which it stands for Euonymus fortunei, which is a ornamental bush. So there are lots of applications for environmental DNA, including looking for specific species. But in our case, we're just looking to rapidly measure biodiversity. So we took a very specific approach to using eDNA, and there are lots of different methods. It is really good for rare species. So I mentioned before, at the beginning of the presentation, that, you know, how do we find those species that we can't see or hear, or, you know, like easily observe? Well, eDNA is great for those, because when the population density is low, so really rare things, and it's hard to survey them. So for example, something you might need a permit for, eDNA is great for that. So, you know, by contrast, if you were doing an inventory of, let's say, hippopotamuses in a pond, you'd have an easy job because you can easily count them. But if you're looking for some very shy newt or salamander or zebra mussel larva, a physical inventory may not work out. As I mentioned at the very beginning, the economics associated with DNA sequencing and the amount of usable data have really driven this. So we ran 4 tests in these 3 ponds, and we generated approximately 320 million megabases. So in 2007, this would have cost about $320 million, and we did this for around $750. So, you know, that's an incredible price drop. And just kind of to put that in context, so NASA today predicts that it would cost about 20 to 30 billion to put a permanent colony on the moon. So imagine 15 years from now, that costing $20,000 to $30,000. So that's how much the price has dropped. You know, really, really unlikely that we'd see those kind of price drops in other industries and areas. So I don't want to overstate what we did, because we did the easy stuff in this environmental DNA. We performed the collection of water, and a DNA company, Jonah Ventures, did the other 7 steps. So they performed the extraction and amplification, indexing, sequencing, bioinformatics. And as I mentioned at the beginning of the presentation, we paid for part 1. So the 4 tests at the top, the numbers in brackets are the gene regions. So for the biologists on the call, those were the gene regions that we targeted with this DNA sampling. The good thing is that Jonah kept our samples, and they're stored at minus 60 degrees centigrade. So we can just send them some money and ask them to run the additional tests and learn some more information. So as I mentioned earlier, we targeted 3 ponds in the Carp Barrens. And for people who aren't familiar with Carp Barrens, it's an interesting location because it's at the top of a hill. So generally the water is just flowing away from the Carp Barrens. So major water bodies like the Ottawa River or minor ones like the Carp River don't have any impact on it. We did choose ponds with different observed characteristics, but they're all kind of have some human presence. So we have Fen Pond, what we call Rockwall Pond, and colloquially known as Peggy's Cove Pond, these 2 locations that we sampled there at Peggy's Cove Pond. These ponds have different water flows, and so, you know, looking at this here, this Peggy's Cove 1 location, we sampled near the outflow, and as we expected, we saw more DNA concentrated there. So, you know, for future, we would look at these hydrogeographic maps to plan our sampling. So the collection day was on October 17th. Wasn't really ideal time of year. Probably June or early July would have been better, but we didn't want to wait. At least I didn't want to wait for another year. So that's Mary Gwen, our biologist. She's at Peggy's Cove and she's extracting some water. And in the middle, I'm basically just pushing the water through a filter at the end. So for this method, it's very simple. We're just pushing the water until the drip rate is 1 drip every couple seconds. So we were only doing between 50 milliliters and 170 milliliters of water collection. So once that's done, we unscrew the filter, we squirt in some 95% ethanol, we close up the filter caps, and we ship them back to Jonah for sequencing. and they send us all the information. So they do those 7 other steps. So about a month later, we had our results. So as I mentioned, late in the year is not great, but also it rained pretty heavily. So it's not ideal because it puts things in the pond that normally may not be there, and dilutes the DNA. And I'll talk about, the things that normally maybe shouldn't be there. I'm happy to report that we didn't find any human DNA in these samples. So that means we didn't contaminate the sample, and also nobody was swimming in these ponds at that time of the year. So just a bit about how I'm going to present the results visually. So if you see a large image like the one on the left, there's more abundant DNA, and the one on the right has less abundant DNA. And you'll also notice this is a nice clear picture of the world's happiest beaver over here, 100% DNA match, the coyote somewhat less. We did focus our results on DNA matches of 97% or higher, but this one in this case would represent 55 to 10%. I specifically chose 4 species per test because we're really talking about 4 nucleotides, 4 letters of the DNA alphabet, A, C's, T's, G's. And just as a reminder, we're not physically observing these species. So let's start off with our vertebrates. So I'm a bit sad to report that the dogs accounted for the highest percentage concentration of DNA in the vertebrate category. So 100% DNA match at 55% of the abundance. The muskrat was 34% of the DNA for vertebrates. And it was found in Fen Pond. Beavers, much less, and the coyotes also much less in terms of abundance. These were both slightly less than 100% matches, and I'll talk about those dogs a little bit more later on. So the next grouping was phytoplankton and cyanobacteria. So phytoplankton is normally invisible, so usually there may be 1,000 of these in a single drop of water. And all of these species were found at 100% DNA match. And the one here in the top left was represented in all 4 ponds, and the others slightly less so. Phytoplankton is super important. It forms the base of marine and freshwater food webs, and it's a key player in the global carbon cycle. So they account for about half the global photosynthetic activity and half the oxygen production, despite being only 1% of the global plant biomass. So super important role that phytoplankton has. Cyanobacteria, by contrast, is sometimes called algae, but it's really actually a bacteria. It's a group of photosynthetic bacteria that they're evolutionarily optimized for conditions of low oxygen. So you hear these things, blue-green algae blooms. Well, it's actually a blue-green cyanobacteria bloom. And again, like phytoplankton, would normally be invisible, but when there's too many nutrients in the water, then you'll have these algae blooms. And cyanobacteria blooms are very problematic because they produce cyanotoxins, which are fatal to vertebrates. So, even in the region, we've had dogs die from swimming in ponds with cyanobacteria blooms. And, we often find our beaches are closed after large rainfalls. Good to note, though, we did only find trace quantities of these cyanobacteria, so in 2 ponds. So even though their relative abundance makes it look high, they were actually just in trace quantities. So that's good news. And we certainly didn't see this kind of scum that you see down here on the bottom left inset. That's just a kind of a picture of an blue-green algae bloom. So macroinvertebrates were our next category. And these can't escape pollution, so they're really a great indicator species. That these can't move from pond to pond. And macroinvertebrates are either small aquatic animals or they're the aquatic larval stages of insects. So that could be dragonflies, stoneflies, snails, worms, beetles. Macroinvertebrates are a good indicator of quality because they're affected by the physical, chemical, and biological conditions of the stream. So they kind of show the cumulative effect of pollution. So we'll start off here first with our rotifers. So the rotifers are like the garbage collectors of the pond. So they're eating dead bacteria, organic, like dead leaf matter, algae, and protozoans. And then we have some amoebas and arthropods were also found in our ponds. We did find fish in all our ponds, and our number one position here is the central mud minnow. The lifespan for most of these fish is between 3 and 7 years, and the mud minnow can survive in low oxygen environments. And we know that some of these ponds almost dry out completely in the summer. So that's not a very big surprise. And most of these fish can actually tolerate very warm water. The brook stickleback and the fin-scaled dace were both found at slightly less than 100% DNA match. You can see that by the faded picture here. And the pumpkin seed wasn't that abundant, but it 100% DNA match, and it's a good indicator species because it has a lot of susceptibility to silt and pollution. Mollusks, we found only one mollusk, but we didn't actually pay for a mollusk test, but we did pick this one up in Fen Pond. And they're intolerant of high salt levels and they're impacted by logging. So again, they'd be a really interesting indicator species. So that kind of concludes what we're going to talk about, the species that we found. So if we look at our ponds in general, we did find that each location is diverse, meaning there's some biodiversity in each location that's not found in the other one. So 28 species in Peggy's Cove 1 that wasn't in Peggy's Cove 2, and so on for these other locations. And there's very little overlap across all 4 locations. You probably noticed that only things like the central mud minnow was in one location and a certain algae was in, all locations. These findings are pretty consistent with some other studies that I looked at. So there was a 4-year study of 425 ponds in Cheshire, UK. And even though an average of only 28 species was found in each pond, a total of 277 species were found across all ponds. So these results aren't inconsistent with what we saw. And if our sampling techniques had been, like I said, slightly better, walking around all the areas of the pond and taking kind of scoops and then putting it all together, it might have improved the homogeneity of our findings. If you really like Venn diagrams, that's actually what things look like. So there are a lot of species still to examine. So we only presented, the top 4 or so in each category. And we did have 672 unique sequences. So we think minimally we're at around 200 species, but we need to make some decisions. And, if you're a biologist and you want to join our group, just send us an email at dna@carphills.com, and, we'd be happy to take your input on how to resolve the number of species. We did have obviously one big disappointment in our findings, which was dogs. I mean, dogs are not allowed in the Carp Barrens, and we found a lot of dog DNA. So that unfortunately is a big bag of dog poop that was found in the spring. And one of the interesting findings was that, dogs were, muskrats weren't. And we know that when domestic pets come into the landscape, there's a lot of predation. So not just, muskrats being shy and not being found, but we also have Kasia has done presentations on Blanning's turtles, and they can be killed by dogs. So and not just that, but there's secondary effects so that you wouldn't think of. So like flea treatments ending up in the water, or pesticides in their food ending up in the water. So doing the sampling that we did after a large rainfall was kind of, a silver lining was that we found a lot of dog DNA, probably as a lot of their urine and feces ran into the ponds. And it's just a kind of a reminder that maybe we need to a suggestion that we might want to improve the way we enforce dogs being there. So kind of looking towards the future, one of the things that I thought would be really interesting was to create some kind of report cards that would help us track how our species diversity is doing, and comparing and contrasting these human visited ponds with remote ponds that are maybe, you know, really off the map in the Barrens or in the Crazy Horse area. Other similar things were indicator species. You know, in terms of our report card, it'd be really interesting to have indicator species. Those are the species that are really intolerant to poor and changing conditions. So there's this ratio called the EPT ratio. So that's, you know, mayflies, stoneflies, and caddisflies. They're Latin names. And looking at, at least in the macroinvertebrates, those species are good indicators. Especially the mayfly larvae, because they have very fragile gills that are very intolerant to pollution. So again, kind of future ideas for these report cards. Similarly, aliens and invasives. So we have the one alien that we're sure of, you know, that was the dog. And conceptually, the idea is that aliens, you found them, but they haven't moved in. Whereas invasives have, you found them, they've moved in, they're breeding, and they're out competing potentially native species. So I think that's spotted crayfish down there. We didn't find any invasive species, but we didn't have the time to look through all the species names and also do the DNA analysis there. I shouldn't say the DNA analysis, I should say we didn't have time to actually look up the species that we found against the invasive species.ca website, for example. So again, kind of where we hope to be. We really would like to see more species in our next run. The northern water snake would be great. Boreal chorus frog in the middle, our landing turtles that we're very familiar with on the far right, a little picture from a bike ride I did. And would be really interesting if we did our bacterial assay, if we picked up, you know, E. coli, for example, in the ponds that all the dog poop is found. That would just be like a further evidence that things are not good when we have these alien species visiting our ponds. So where to from here? So, you know, this was a very informal pilot. It would be great to consider larger, more formal projects in the future. You know, my report card kind of indicated something like that. We do need support from biologists prior to carry on and to apply for additional funding. So, you know, doing good science here. You know, that report card would be really cool. You know, monitoring kind of ponds that have been visited by people and the remote ones, for example. So how can you get involved? So we have some easy ways for you to get involved. So you can just join our eDNA working group. So just email us and just say, hey, I'd like to get involved, and we'll go from there. You can test your knowledge of eDNA. You can take our quiz. I think the link to it will be on our Carp Hills webpage. If you want to donate, if you're interested, so we can finish part 2, it's going to cost around $800, and we can maybe use any additional funds for remote ponds. So there's a donation link on that page as well, which I'll show you. If you're just interested in general, you know, testing your own lakes and streams, the places that you love, you can buy a kit from a commercial company. They cost between, you know, $50 and around $500, depending on the number of tests you want done. You can do really simple ones like just fish, for example. And you can just learn on your own. You can visit our webpage, and we have links there to all the bioinformatics, the whole DNA pipeline, what primers were used, the species, word cloud, and some other interesting stuff as well. So I just want to summarize, we cast our net really, and we found quite a few species, and we hope we aren't done yet. And we hope you'll join us on our next stage of our journey, and see what you can find in part 2 and beyond. So thank you very much for joining us.
Well, thank you, Mike, on behalf of Friends of the Carp Hills. And I would like to say that all the information about the project and the links and the donate button and the data is on the Friends of the Carp Hills website. And if you go to the top menu and look under our initiatives, do the drop-down menu under the Our Initiatives, you'll see the Carp Hills eDNA project, and you'll be able to get to the web page from there. So thank you, and have a good evening.